DNA is a microscopic string of information stored in every cell of your body. It consists of just 4 letters, repeated over and over again, that make up a unique genetic blueprint. Scientists all over the world have collaborated to discover the exact sequence of those 3 billion letters. 3 billion letters. If you tried to read every letter in the human genome end to end, it would take you almost 300 years. But a computer can do that much more quickly. I write programs to compare unique genetic blueprints. This makes it possible to discover the secrets of humanity. One recent example of comparing unique genetic blueprints is between the Tibetans and the Han Chinese. The Tibetans live on the harsh, cold, high-altitude Tibetan plateau, while the Han Chinese live in the same region, but at a much lower altitude. What can their genes tell us about the secrets of survival? Well, the problem is you can't directly compare one person to another person. Each person has some individual variability, and the same is true of their genes. If you want to compare the Tibetan population to the Chinese population, you need more samples. More samples means more data, which means you need programs and computers to sort through it all. So let's take a look at what that might look like to compare those 2 consensus sequences. So up here you have your Tibetan consensus sequence from many Tibetans and your Han Chinese consensus sequence from your many Han Chinese. That gets input into a program, some code that looks like this. So you can imagine me sitting at my computer typing away. This is actually some code that I wrote to generate something like this. And then you get the output, which is the difference between those 2 consensus sequences, visualized here with that little green segment. After such an analysis, it was found that there is a difference in the gene for hemoglobin. Hemoglobin is the protein in your red blood cells that binds oxygen in your lungs and carries it all throughout the rest of your body. You can see how that would be important in environments where oxygen is sparse, such as the high-altitude Tibetan plateau. Humans are so diverse. To discover more defining genomic variations, like the Tibetan hemoglobin example, we have just a massive amount of data that we need to sift through. My graduate work is focused on writing programs to understand this tapestry of human variation and existence.